Hi, everyone. I am here today with Jack O'Mara, who is the founder and CEO of Ochre Bio. Jack, thanks for joining me today and welcome.
No problem. Nice to meet you. Thanks, Neil.
Yeah, my pleasure. So today we're going to talk a little bit about your company and what you're building. So Jack, why don't I turn it over to you to give an introduction about the company, founding team, and technology, and then we can dive into some Q&A.
Great, Neil, that sounds great. What I'll probably do is I'll talk you through at a high level what it is we're trying to achieve. I can give you a little bit about the background story and how we've gotten to where we are today, and then maybe talk about the genesis of Ochre Bio and how the team and how we got together, and then pause there. We can dive into Q&A and see where you'd like to take things or where it might be most interest to your audience.
That sounds great.
At a high level, Ochre Bio is a deep phenotyping company developing RNA therapeutics for chronic liver diseases. Chronic liver disease is the only top 10 global killer that's currently on the rise. We have built one of the largest genomic atlases of the human liver and used it to uncover a host of novel therapeutic pathways that we've been busy validating in our labs here in Oxford, UK. And one of the things that's a little bit different about our pipeline is that once we find a target and developed an siRNA therapeutic, we actually go directly to testing that in a whole human liver that we keep alive on machines. The thesis being we want to keep our pipeline human relevant from genomics-based target discovery, in vitro validation, all using primary human hepatocyte models, and then actually testing out therapies in the liver itself. We do mice workups, but really for conviction, it's the actual human organ itself. And that's the first indication we're looking at as a company. Our first program is in transplant, so essentially trying to improve outcomes for the use of high-risk donor livers, which is increasingly a challenge in the transplant space. But really, we see this as a first indication in a wide portfolio of products our company is very focused on developing. And we think of ourselves as a chronic liver disease company rather than a transplant company. A little bit like how the gene therapy companies think about orphan indications or rare genetic disease. Transplant has a lot of the same regulatory benefits, and ultimately, we can make a big impact in this space that helps us segue into larger and larger liver-related diseases.
Yeah, Jack, sorry, no, just one question for you. Going back to this concept of deep phenotyping, which is sort of the core of your technology, could you explain a little bit about what that means?
Yeah, absolutely. Why don't I pull up a slide here to help illustrate what I mean when I say deep phenotyping, because I know it is a funny term. But essentially, deep phenotyping is the marriage of genomics, spatial sequencing, imaging AI, and genetics and machine learning to try and tie together all of that data. So one of the first big projects we completed as a company was this genomic atlas I mentioned of 1,000 human livers. And what I'm showing you here on the left is two biopsies of the livers that were included in that study. And essentially what we did was we took biopsies of every liver that was transplanted in the UK in 2017 and 2018. And we did a full imaging AI workup. So we did about 6 or 7 different stains and imaging to try and standardize the histopathology of these livers, because histopathologists won't agree with themselves from morning to noon. But that helped give us a baseline for the actual disease state that these tissues were. And then we used a spatial sequencing technology to annotate different disease niches. So what I'm showing you here in blue is a particular disease niche of interest to us. It's essentially the beginning of inflammation or ballooning degeneration is the name of the phenotype. So we use an imaging AI tool to annotate regions of particular disease stages, and then use a spatial sequencing tool to take samples from that particular region and sequence them and compare what was different on a transcriptomic level between a particularly disease stage and a more banal steatosis, more banal fat stage, or even healthy liver tissue, and compare that across livers, compared that across 1,000 patients, and then ultimately compared it to 150 clinical variables we had on all of these patients, because the NHS in the UK is great for really rich phenotypic information on these patients. So we're able to understand how this related to obesity, their bloods, all of the UK biobank data, and ultimately come up with interesting hypotheses around what are the targets that are driving the progression of the disease, or what's changing as the disease progresses that helps us ultimately come up with clues about what might be the best therapeutic or best ways to intervene therapeutically. And one of the things that was a bit unique about this particular study of as it relates to chronic liver disease over 1,000 patients was that we focused on earlier stages of the disease. So in the context of NASH, a lot of people are very interested in NASH right now. But oftentimes companies will look directly when it's quite inflamed at the very end stage of the disease where people will take biopsies and as a result, you only see a very small spectrum of what is a 15 to 20 year disease that's been playing up over a long time. So our focus on transplant allowed us to study patients from healthy all the way through to fibrosis. So we have this really wide spectrum of disease stages to help us kind of infer or come up with novel therapeutic targets that we were ultimately able to then move into validation and develop siRNA therapeutics around, et cetera. So anyway, I hope this illustrates what we mean when we say deep phenotyping. It's essentially getting rich phenotypic data that helps inform what's actually going on in the silent years of chronic liver diseases.
Yeah, and Jack, just to sort of dive into that point a little more. So, if I'm understanding things correctly, I mean, you're amassing a tremendous amount of data in this deep phenotyping or phenomics type approach. Can you talk a little bit about how the sort of collection of this sort of big data and all the data related to liver disease helps informing and sort of build a competitive moat around what you're doing in terms of translating that to drug development?
Yeah, absolutely. One of the things that's kind of funny is we talk all about the sexy computational approaches and machine learning, but really you're kind of hitting the nail on the head that it's the amount of tissue and amount of data we can generate that is the key to the success. So it's what that translates into is me knocking on doors of transplant surgeons' offices all around North America and Europe to try and get access to discarded donor organs as a means to really build up really strong data moat in a way around these indications. And we've been doing that. We're now operational in 6 transplant centers. So UCSF over in Europe neck of the woods, UMass, and a few others in the US, as well as Birmingham, Oxford, and a couple others here in Europe, where we take livers that have been discarded and take different samples at different time points as we keep them alive for multiple days in these machines to really build up this kind of in silico liver model that helps us inform some of the target discovery work we do. And then the beauty of RNA medicine, as the world has realized now with COVID, is that you can go so quickly from a sequence that you think is really relevant therapeutically to actually developing a fully optimized RNA therapeutic candidate. And that's what we've been able to set up the infrastructure in Oxford to do, is to go very quickly from seeing signal in our data to identifying the target to optimizing which potential RNA candidate was most relevant, to designing that and then testing it in a whole human liver. And that kind of speed from target to therapeutic is, I think, what is a lot of ways our defensibility or our kind of USP, as they like to say.
Yeah, and I love that approach, right? I mean, you're using human liver tissue as the sort of primary early stage, source to validate your therapeutics, as opposed to typical in vitro, in vivo animal models, which, as we all know, don't necessarily translate all that well into the clinic. So I think that's a huge differentiator. Jack, I want to transition a little bit to the team. And you're based in Oxford in the UK. Could you talk a little bit about your founding team, some of your advisors, how you all came together to work on this problem?
Yeah. I can give you maybe a little bit of the genesis story of how we kind of got started as a way to form the team we started to build together, and really excited about the folks we have working at Ochre Bio now. But really the work came out of our CSO's 20-year history in liver drug development. He was originally a medical doctor before the turn of met him in South Africa. But then when the Human Genome Project came online, like a lot of geneticists at the time, flocked to London, where all of the excitement was at that point, and really retrained in computational biology and systems genomics, and ultimately went on to co-steer Oxford University's Single Cell Genomics Consortium before taking the job of head of genomics for Novo Nordisk, which is the big diabetes company that everyone knows. So they were really interested in NASH, which is a big thing. Still is, but has kind of hit a few walls, clinical trial bottlenecks. But the big challenge for someone like him doing a lot of the target discovery that we do at Ochre Bio at Novo was that it's very hard to see a clinical translation, or what's the route to actually impact, because your preclinical models are terrible. So you can do all this genomics, but then you got to go into a DB mouse, and that's terribly unsatisfying if you've done all the work, just because of how poorly predictive those models are for actual chronic liver disease. But equally, for an indication like NASH, your translation is very difficult. You don't have very clearly defined endpoints. The regulator still hasn't really decided what they want to see. You also have to run these massive trials because it's a silent disease for so long. Not dissimilar from neurodegenerative disease like Alzheimer's. Patients don't know they have the disease until it's very advanced, and then as a result, your studies need to be huge and multi-year. So when companies like Novo are nervous about it, we were starting to think about what could we do differently. And that was when we had a dinner with the head of transplant medicine at Oxford, who had just created these new devices. They made the front cover in Nature, these perfusion devices that take donor livers, keep them alive outside the body. And surgeons really invented them to better study these livers before they go into patients, to make sure they're safe for patients. But it was really an interesting aha moment where, wow, we can actually study these livers on a machine as a preclinical model. And that solved that first problem of mice models, because you could actually study the human physiology, perturb them, sequence them, really understand if you're having a therapeutic effect. But the reason I got involved, and the reason we spun this out as a company rather than continue to do this work at Novo, was that there is a real line of sight to a clinical trial as well. We could run a tractable clinical trial on transplant, for a small company like a brand new biotech can afford to do that. It's relatively short endpoint. So the primary endpoint is liver enzymes 1 week post-transplant, which we know we've got good handle on, and the regulator is very au with. And then you can also ultimately use this data to segue into whether you're having a broader systemic effect on the patient through a delivery of the therapeutic ex vivo. So that idea of having a full stack biotherapeutics company where we can do the target discovery, we can develop the therapies, and we can go to trial in a cost-effective way for a small company to ultimately de-risk much larger, much more difficult indications like NASH or chronic liver diseases more generally. That was a really attractive proposition to get started. So my background, I was originally in biomedical engineer, but ultimately went on to spend a number of years in the US working for early stage health tech and biotech companies. Got very fed up with it. Again, Europe's kind of lack of translational focus, where I studied in Ireland. You might have picked up from the accent. And ultimately ended up at a group called Avexis, which was one of the first AAV gene therapy products to get approved and help get that over the regulator, over the finish line. Before coming back to Europe, I wanted to get closer to home. I was starting to get an American accent, which was making me nervous. So I wanted to get back across the water. And then met Quinn and heard all about the liver and his research. And really, we've been all go ever since. It's been a really exciting time. We brought in, because we were based in Oxford, we managed to very quickly pull all of Quinn's old team from Novo into the company. So within a couple of weeks after financing, we got about a lot of the top NASH scientists come over to our group. And Oxford is a real treasure trove of just incredibly smart and driven people, particularly scientific people. So we've been really lucky with building out our R&D team there. And we just hired our first, well, not just about 8 or 9 months ago now, we hired our first US employee, a guy who's been leading our transplant partnerships there. And he was formerly CEO of a group called Essential Pharmaceuticals, who sold organ preservation solutions at transplant centers. So been a great kind of asset to the team to help us get partnering up with some of the major institutions over in the US as well. So, and I should mention, we also opened up a Taiwanese group where we do a lot of our target discovery now in Taipei, and brought on a strategic investor from Taipei to get access to some of the hospital systems there. So we're a very international group, but really have a strong team coming together.
That's great. And, I was just circling back to your days at Avexis. I mean, that was, I think, the first gene therapy that was approved for spinal bifida, if I recall. And I think that it was a nice outcome that they were acquired by, was it Novartis for a couple $1 billion, I think, a few years ago.
$9 billion.
$9 billion. Okay, more than a couple. But yeah, so a nice outcome, and probably very nice to see how that was actually translated to a commercial product. So probably a lot of learnings there that you're applying today.
Exactly.
I know I want to be conscious of time, but just probably one more question for you. I always like to ask this. What do you sort of see as your most near-term risks or obstacles that you need to overcome to sort of de-risk the technology, de-risk the path to the clinic?
Yeah, I think for us, a lot of the core science, we've been able to stand up quite quickly, but I think the big risk in a lot of ways for us is being able to get access to liver tissue, which is kind of our key input for the type of development we do. I know it's a silly analogy, but sometimes we like to compare it a little bit to the Tesla's lithium problem when they got started. Ours is the liver problem, but we've been very quickly and very actively partnering up with transplant centers around North America and Europe. But I think being able to continue that on that thread and make sure we have enough of the relationships and the infrastructure in place to test all of the targets that are coming out of our target discovery group will be key to making sure we're able to capitalize on this kind of novel approach and this new perfusion technology that makes it all possible.
Yeah, that sounds good, Jack. Well, I appreciate your time today. I think we could probably literally talk for the next 2 days about your company and technology, but in the interest of time, I will let you run, and appreciate your time today.
Thank you, Neil. Really great speaking with you.